Hi, everyone. My name is Ariel Nicastro, and my research project is acoustic forcing, device design and applications in neuromodulation. I was working with Corey Herr and Professor Wolfgang Losert. I'm from Middle Tennessee State University, and in partnership with UMD Physics Department, Institute of Physical Sciences and Technology, and the Losert Lab. So let's get started. One second. There we go. So acoustic forcing is the ability of mechanical waves to interact with objects in its path. This is analogous to the background of my research project, which is focused ultrasound. So what is it? It is essentially focused energy used to non-invasively interact with targets in the body. There are two main types of focused ultrasound. The first type being high intensity focused ultrasound, known as HIFU for short. And you can see an example of this on the right side, as it is used in the human body to target larger biological objects, such as tumors, for example. So on this left side, you can see an ultrasound transducer that essentially emits the ultrasound, and these ultrasounds focus inside the tumor, and the goal is to get rid of it. So there's also low intensity focused ultrasound, which can be used in the human body in a similar way, where you have a transducer by the human body. And this is related to neuromodulation, which I will discuss right now. It is the use of LIFU to non-invasively stimulate neural networks in the body. And it has been known to stimulate neurons both in cells and in live animals. A study has been shown to modulate behavior in monkeys. It modulated their visual motor behavior primarily. So one goal for neuromodulation in humans is to treat disorders such as epilepsy, chronic pain, and migraines. So over here on the right side is basically some human neural progenitor cells, HNPCs, which are stem cells that act like neurons. And this was imaged by the Losert Lab. So let's get into my research project. My goal is to design an ultrasound forcing device that can successfully pattern microbeads. So where did the microbeads come in? If I'm able to pattern microbeads, it shows that this patterning will make sure that it can be used in future experimentation on cells and neural networks. So here's my device. Over at the top, you can see a diagram of my device, and then at the bottom is the actual version. So the first step in creating this device was to 3D print a hollow structure. This is 22 by 22 millimeters, so quite small, and it is made out of a biocompatible resin, so it can be compatible with these neurons. And then on all four walls are glued piezoelectric ceramic plates, and these plates essentially vibrate, creating frequencies that will represent the focused ultrasound. And the next step was to solder stranded silicone wires to the front and back of each plate. These plates operate with an AC circuit, so this configuration of wires allows for the plates to vibrate. Then the third step was to connect the wires to a function generator. And this is where I can change the frequency and the voltage of the waves being emitted from the piezo plates. So how does this device work? This device is quite small, as I previously said, so it can fit into a 35 millimeter Petri dish, which is what we use in the lab. And opposing piezo plates will create standing waves, which is how I was able to test my device. So over on, let's start with what are microbeads. Microbeads are about 20 micrometers in diameter, a similar measurement scale to cells. And when I had a 2 megahertz sine wave coming out of two opposing piezo plates, they created this pattern of waves that you can see here called standing waves. There are sections of activity called antinodes, and then there are quiet areas in the center called nodes. And these nodes are particularly important for this test because when this device is in the Petri dish, we have water, and these waves are propagating through the water. And when I drop down microbeads with a pipette, these beads will settle down into the nodes of these waves. And you can see that this is actually a side view of the experiment. They will settle into lines at the bottoms of the Petri dish. So this is important because if the frequencies do create these lines, there's proof that the frequencies will also propagate all throughout neural cells in dishes. So here's a top view of my microbead patterning, and this was imaged with the help of Anna from the Losert Lab. So you can see from the arrows that these are the sources of acoustic forcing. And these lines are essentially what you see, which is representing the nodes of the 2 megahertz sine wave. The way to quantitatively test that my device works is to measure and take the mean of the spacings between the lines. So this gave me a 350 micrometer spacing. And this was a theoretical version. So the actual calculations that I calculated was 338. So that is quite close. And in conclusion, that proves that my device is successful and frequencies will propagate through the medium. So the next step is to stimulate activity spikes in neural cells. This is something that we are in the process of doing. And we have used trials of both primary neurons that are derived from rats and human neural progenitor cells, HNPCs, which once again are the stem cells that have neuron-like tendencies. So you can see in this larger image of the dish that over time, the brightness of the cells will decrease. And this is a small section of what is happening all throughout the dish, but you can see it enlarged over here on the right. And in the circle is one specific neuron. This is one of the many neurons in the dish that have this activity. It'll decrease in brightness rapidly. So rather than a gradual diminishing in brightness, this quick diminishing shows that the cell activity, the cell is dying essentially. So the results show that the frequencies used were too powerful for the cells to survive. So what do we do from here? The next steps is to reduce the power of the ultrasound forcing by lowering the voltage in the function generator. And then, of course, we will continue attempts to stimulate activity in neural cells. And another potential activity that you can do with the acoustic forcing device is to pattern neural cells, which will essentially provide us with more information about how the cells will respond when they're already plated in the dish rather than being inserted into the dish like the microbeads. So in conclusion, I created an acoustic forcing device that was designed to replicate the effects of focused ultrasound on neural cells in a petri dish. My device successfully patterned microbeads, proving that it will also propagate waves through petri dishes of neural cells. Tests on these neural cells showed that some of the cells died, so future trials will have waves of lower voltage. And as an acknowledgement, I would like to thank the Trend REU program, LoCERT lab members, and Dr. Daniel Serrano for a fantastic summer opportunity. Thank you for your time.